Right, hello folks, welcome to the final lecture in the experimental design and analysis section of the core skills modules. So I say final lecture, it's the final bit of new taught content. We will have another wrap-up session soon on live online. I will also run a revision session much closer to the exam, which will be in June. So by the time we get there, it will have been a long time since you've actually seen the lectures in the first place. So we'll have a recap then to try and refresh your memory. So today's job is to go back to thinking about trends. For much of the sort of second part, at least, of this module or this section of lectures, we've talked quite a lot about testing for differences in various different ways, using things like t-tests and analysis of variance. And we've only really, I think, in one lecture touched on the idea that you can also often you're looking for trends in data sets. So we looked at correlation as a way of approaching analysis of trends. Today I want to return to that theme, but from a slightly different angle, and look at a concept called regression, and then also something related called analysis of covariance. The gratuitous Ugg boots on this slide are there because I needed an image to reflect a trend. This is like going back a number of years now when Ugg boots were very fashionable, and I was a bit surprised because, having spent some time in Australia where Ugg boots are generally associated with the least cool people on the planet, I was a bit surprised that they were ever fashionable in the UK. I sort of every year that went by, I became ever more stunned that they were still fashionable. So I promised myself I'm going to leave it on this slide until such time as somebody in my lecture class does not own a pair of Ugg boots. And last time I had had the cohort in a real classroom, there were still plenty of people who had Ugg boots, so they're staying there because apparently the trend lives on. Anyway, the point is it's about trends, this lecture, hence the irrelevant picture. Right, so the outline for today's session is to look at trends, in particular when there is a possible causal relationship between the x and y variables, the dependent and independent variables. And this is going to lead to the idea of linear regression, which is a form of analysis that we use when we're looking for a causal relationship, when we think x is causing y to change. So that's the main topic really for today. And we're going to look at how linear regression works, and also how we can use it to make predictions about what you might find in the future. And inevitably, because this is a parametric statistical test, we will look at the assumptions of regression, but they won't look too unfamiliar. They're very similar to the kinds of assumptions we've made in other parametric tests. And then finally, in the last section of the lecture, it'll be a lecture in 3 parts as normal. In the final section, I want to look at what happens when you have both continuous predictors, as is the case when you're looking for trends, but also categorical factors, like when you're doing analysis of variance. So what happens when you have both types of predictor in the same analysis? And the answer is something called analysis of covariance happens, or at least that's often the way that we analyze the data. So we'll look at analysis of covariance, which is a kind of hybrid of analysis of variance and linear regression. And because it's a hybrid, it looks very familiar to you once you've learned how analysis of variance and regression work. Okay, so lecture in 3 parts. The first part is going to focus on the sort of concept of regression, and we'll start to look at how regression works. So, in a previous lecture, I talked about Pearson's and Spearman's rank correlation being good ways for testing for trends when you aren't sure if there's a causal relationship between the 2 variables that you have, or you'd think there isn't a causal relationship, but you're nevertheless interested in looking for patterns. So we talked about the example of looking for a relationship between the number of storks arriving in Denmark and the number of human babies being born. We don't expect that storks cause babies to be born, or indeed that babies cause storks to arrive. But that doesn't mean that we're not potentially interested in any pattern between those 2 variables. So in that sort of situation, a correlation analysis is appropriate. But sometimes, quite often actually, especially in experimental settings, in laboratories for example, when you're manipulating one variable and looking for a response in another, let's say you're changing temperature and you're looking to see how growth rate responds, in those situations you expect a causal relationship, you're interested in a causal relationship, and in those cases linear regression is the appropriate test to use. Correlation would be okay, but linear regression is a bit more powerful, a bit more flexible, and it tells us a little bit more about what's going on. So that's what we're going to look at today, linear regression. And it's a parametric test, just like analysis of variance, for example, that we dealt with in the last two lectures. And actually, it works very similarly to analysis of variance. In fact, you create an analysis of variance table, an ANOVA table, in order to perform your regression analysis. So we're going to do that today. And helpfully, it uses the same test statistic, F, as analysis of variance. So it's going to look very familiar once you've got your head around the last two lectures. And of course, inevitably, the objective is to calculate a test statistic which quantifies the likelihood, which reflects the magnitude of the difference between what you'd expect at random under the null hypothesis and what you've really seen. And from that, we calculate P, the probability that the difference you've seen between your sort of null expectation and the reality that you've got in your real data set, the probability that that could have arisen by chance. And if we think it's very unlikely to have risen by chance, we have a very low p-value. In that case, then we would conclude that actually the null hypothesis is likely to be wrong and something more interesting is going on in the form, normally reflected in the form of our alternative hypothesis. Okay, so it's a bog standard stats test. In other words, we're going to take data, use the data to generate a test statistic, use a computer to tell us what probability goes with that test statistic, and hey presto, we'll have an answer. Along the way, there actually is going to be another statistic that we're able to calculate from this regression called R squared, sometimes lowercase r, sometimes uppercase r, but it's the same thing. It's kind of related to the correlation coefficient R as well, but it is R squared is a really useful way of quantifying the strength of a relationship that you've detected using a linear regression analysis. So it's easiest to understand these things working with a real example or a kind of simulated example. So this comes, I've made up some data, but it comes from a real study, or it's based on a real study of Drosophila fruit flies, which is kind of every biologist's favorite study organism. Drosophila is this incredibly well studied, well understood tiny insect. Often in the life sciences, when we talk about insects do this, we're really talking about fruit flies, because most of the work's been done on fruit flies, and it's because they're really easy to work with in the lab, and they have some basic properties which make them useful for studying everything from molecular genetics to behavior and ecology via biochemistry and neuroscience, by the way. So everybody's in on the act here, although it's hard to see how fruit flies are relevant to sport and exercise science, but I'm sure there is a way. Anyway, for the purposes of this lecture, I'm going to use fruit flies to teach us about regression. And we're going to look at an experiment that should be close to the hearts of all of you, looking at the relationship between diet and longevity. So longevity is the posh word for how long you live. And humans, of course, are obsessed with living longer. Some people want to live forever. And some experimental data coming out of Drosophila, and also from similar experiments with rats and other species, and even some data from humans, have suggested that diet can be quite closely related to longevity in a way that isn't necessarily immediately intuitive, and specifically that restricting calories, restricting the amount of energy available in food, might create the conditions in which individuals live longer. So almost like counterintuitive, really, that you might starve an organism or sort of partially starve it, and it might actually live longer as a result. So we're going to look at some data from Drosophila from an experiment that looked at this hypothesis, this possibility. So the null hypothesis in this experiment is that there'll be no relationship between how much energy we give fruit flies in their diet and how long they live. And this is a kind of classic experiment where we manipulate what's on the x-axis, the independent variable, and we look to see how the dependent variable, the y variable, in this case longevity, responds. So we expect a causal relationship between these two things because we're doing a manipulative experiment in which we're changing x. It's the only thing that is changing experimentally. All the other conditions are identical for the fruit flies. We're in charge of manipulating the energy content of the food, and if there is any change in longevity, it must be as a result caused by the manipulation that we make to the x-axis. So in other words, there's no way that changes in longevity in this experiment could be causing the food to vary in calorific value, because we're in charge as scientists, as experimentalists, of the diet. So the null hypothesis is that there's no relationship between these two variables, and then we collect some data, and here are the data on screen. It looks like there might indeed be quite a strong negative relationship between the energy content of the food and how long these fruit flies lived. Fruit flies don't live for very long, but they can live for a number of weeks, and so we've got the maximum longevity up here in the order of 60 days. But it seems that when the Drosophila are fed higher quality food with more calories per gram, more joules per gram, we see the fruit flies are living substantially less, it would appear. But there aren't very many data points here. What's that, about 9 data points? So if we imagine throwing darts at a dartboard at random, we could get that pattern just by chance. The question is, well, what is the chance of us getting that apparent negative relationship just by chance if the null hypothesis is really true? The null hypothesis says the energy content has no effect on longevity. So we need to quantify that probability P, and we're going to do it using linear regression analysis. So regression is going to tell us whether or not this relationship is significant. And the way that regression works is it looks for the dependence of the y variable on the x variable, the independent variable. So I've used those words a few times already in this lecture, so just to remind you about this, you've probably encountered these terms before, but the variable on the y-axis in a causal relationship is known as the dependent variable. It depends on what happens on the x-axis, which is known as the independent variable. Independent, if you like, because as a scientist and experimentalist, we are free to manipulate the values on x and see how y responds. So the idea is that we choose x, but y is dependent on what happens to x. So that terminology is worth getting your head around because it will crop up quite a lot, you'll find. So in our case, longevity is the dependent variable, and it depends, we think, on how much energy is available in the food. So that's the independent variable. And linear regression, first of all, starts, like with all stats tests, with a null hypothesis that says there's no trend between the variables. And it's helpful, and indeed this is what happens mathematically in the test, it's helpful to imagine that null hypothesis as a function on the graph. So I've drawn a line on the graph to represent the null hypothesis, which is that there is no relationship between the energy content of the food and how long the flies live. And the typical way of expressing that null hypothesis mathematically is to say that the y-axis, the average value of the y-axis, longevity in this case, is constant with variation on the x-axis at the average. So the average longevity for my data set here is about 40 days. And so what I'm saying is that flies on average are going to live for 40 days, whether we give them really low energy content food or whether we give them really high energy content food, which gives us this flat line at about 40 odd. So what I'm saying is that the reason the line is there and not up here or down here is because that's the average longevity of the whole data set. And if the null hypothesis is correct, any deviation from that expected value of about 40 days, so for example this fly here which is living much longer, that deviation must just be random noise. It's nothing to do with what's going on the x-axis, according to the null hypothesis. So there's a single mean longevity regardless of the energy content, and any spread of values around that mean just reflects random noise, or what we might call error or residual variation in longevity, caused by the fact that every fly is different. So each fly in the data set will have a different genotype, it will have maybe experienced slight differences during its development in the conditions that it experienced, and all those things are likely to give rise to variation in longevity, but it's irrespective of the quality of the food provided. Okay, so once you've got your null hypothesis described as a line or mathematically in your data set, you can then measure how well the data fit that hypothesis by looking at the differences between the data and the line. So we can measure the distance between each data point and the line, and that will tell us how close the line is to the data, essentially. So what we do is we calculate the sum of squares for this null hypothesis, because obviously if we just added up all of these differences, we've got some positive ones and some negative ones, and they would kind of cancel each other out. So what we're going to do is square them and add them up before quantifying the total sum of squares for our null hypothesis. So this should seem very familiar, because this is just exactly what we did right back at the beginning of these lectures when we were calculating the sum of squares in order to quantify the variability in the data set. And that's really all we're doing here, is we're quantifying the variability in the data set, in this case in the context of the null hypothesis, which in this case just is the mean longevity. You'll understand in a minute why we're doing this, because we're going to contrast it with a line of best fit when things will change. So the total sum of squares, which is the sum of the squared differences between each data point and the mean, is going to be our starting point for building an analysis of variance table like we did in the previous lectures. But this time, critically, something's going to be a bit different. What's going to be a bit different is we're going to compare that sum of squares to what we get when we have an alternative hypothesis, which is reflected in a line of best fit through the data. So in linear regression, the clue is in the name linear, we assume a straight line relationship between x and y, and we fit a line of best fit through the data points, the best possible fitting sloped line, to represent our alternative hypothesis, which is that there is some kind of trend between energy content of food and longevity. So the alternative hypothesis says there is a relationship, the null hypothesis says there isn't, but mathematically we can be more specific about the alternative hypothesis, and we actually form, we create a line of best fit which has an equation which we'll come back to later on, which represents the exact relationship between x and y that we think might be going on. So it's our best guess, this line of best fit, for exactly what we mean when we say there is a trend between x and y. And in the case of my data set, that line of best fit has a negative slope. So as energy content goes up, longevity comes down. So just as with the null hypothesis, we can calculate the difference between the data points and this line of best fit, calculate those differences, sum them up, and we've now got a new quantity, a new sum of squares to compare against the sum of squares in the case of the null hypothesis. So the difference between the data point and the line of best fit tends to be known in regression as a residual. I've used that word already when talking about the differences between data and the model or the mean in something like a t-test or analysis of variance, and linear regression uses the same principle. Basically, the residual variation, when you've put a line of best fit through your data, represents the bits of longevity, in this case the variation in longevity, which is caused by things other than the energy content of the food. So if the alternative hypothesis is correct and the energy content of the food does affect longevity, then the line of best fit represents our best guess as to how it affects it. But even when you take into account that line of best fit, there are going to be some deviations from that line of best fit which reflect other causes of variation in longevity, such as the genetics of the fly concerned, or exactly which parent it had, or which test tube it was put in the lab. So we got the residual variation, and we want to quantify that in exactly the same way that we did for the sort of null hypothesis, and so we calculate a sum of squares called the residual sum of squares, which represents the remaining variation left over once you've fitted a line of best fit through the data. Inevitably, the residual sum of squares will be smaller than the total sum of squares, because you've put a line of best fit through the data instead of just having the mean. And for any data set, the line of best fit will always be at least a bit better, even if it's only a bit better by random chance, and that's obviously what we're going to go on to assess using a p-value. So the question is going to be, is the residual sum of squares, the bit left over once you've put a line of best fit through the data, significantly smaller than the total sum of squares? So much so that we conclude that the alternative hypothesis is demonstrably a better fit to the data than the null hypothesis. The null hypothesis being that the average longevity is the same regardless of energy content. Another way of saying the same thing is what we're asking here is the trend between x and y accounting for a significant amount of the variation in the y variable. Okay, so that's the end of the first part. We're going to start to put those principles into practice by generating an analysis of variance table for a regression in a moment. So we'll do that after the break.